This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is nucleotide excision repair? Answer: nucleotide excision repair is a DNA repair mechanism in which specific endonucleases remove the oligonucleotides containing damaged bases, DNA polymerase and ligase fill and reseal the gap, respectively. This repair mechanism repairs bulky helix-distorting lesions such as pyrimidine dimers. It occurs in G1 phase of cell cycle and is defective in Xeroderma pigmentosum. Question: what are the symptoms of Xeroderma pigmentosum? Answer: Xeroderma pigmentosum presents with dry skin, photosensitivity, and skin cancer. Question: what is base excision repair? Answer: base excision repair is a DNA repair mechanism in which base-specific glycosylase removes an altered base and creates an AP site. One or more nucleotides are removed by AP-endonuclease, which cleaves the 5' end. AP lyase cleaves the 3' end. DNA polymerase-β fills the gap and DNA ligase seals it. This repair mechanism occurs throughout the cell cycle and is important in repairing spontaneous or toxic deamination. Question: what is the acronym for remembering the key steps in base excision repair? Answer: gel please. Question: what is mismatch repair? Answer: mismatch repair is a DNA repair mechanism in which mismatched nucleotides in the newly synthesized strand are removed and the gap is filled and resealed. This repair mechanism occurs predominantly in the S phase of the cell cycle and is defective in Lynch syndrome. Question: how does nucleotide excision repair repair bulky helix-distorting lesions? Answer: nucleotide excision repair uses specific endonucleases to remove the oligonucleotides containing damaged bases, then DNA polymerase and ligase fill and reseal the gap. This repair mechanism is able to repair bulky helix-distorting lesions such as pyrimidine dimers. Question: when does nucleotide excision repair occur in the cell cycle? Answer: nucleotide excision repair occurs in the G1 phase of the cell cycle. Question: what is the defect that occurs in Xeroderma pigmentosum? Answer: Xeroderma pigmentosum is a defect in the ability to repair DNA pyrimidine dimers caused by UV exposure. Question: what is the function of base-specific glycosylase in base excision repair? Answer: in base excision repair, base-specific glycosylase removes an altered base and creates an AP site. Question: what are the steps of base excision repair? Answer: the steps of base excision repair are: a base-specific glycosylase removes an altered base and creates an AP site. One or more nucleotides are removed by AP-endonuclease, which cleaves the 5' end. AP lyase cleaves the 3' end. DNA polymerase-β fills the gap and DNA ligase seals it. Question: what is the function of mismatch repair? Answer: mismatch repair is a DNA repair mechanism in which mismatched nucleotides in the newly synthesized strand are removed and the gap is filled and resealed. Question: when does mismatch repair occur in the cell cycle? Answer: mismatch repair occurs predominantly in the S phase of the cell cycle. Question: what is the defect that occurs in Lynch syndrome? Answer: the defect that occurs in Lynch syndrome is a malfunction in the mismatch repair mechanism. Question: what is the purpose of DNA repair mechanisms like nucleotide excision repair, base excision repair, and mismatch repair? Answer: DNA repair mechanisms like nucleotide excision repair, base excision repair, and mismatch repair are essential for maintaining the integrity of the genetic information in cells. They work to identify and correct errors and damage in the DNA, allowing the cell to continue functioning properly and avoiding the harmful effects of mutations. Question: what is the difference between nucleotide excision repair and base excision repair? Answer: nucleotide excision repair and base excision repair are both DNA repair mechanisms, but they differ in the types of damage they repair and the enzymes and processes used. Nucleotide excision repair is used to repair bulky helix-distorting lesions such as pyrimidine dimers, while base excision repair is used to repair small, specific types of damage to a single base in DNA. Question: how do nucleotide excision repair, base excision repair, and mismatch repair work together to maintain the integrity of DNA? Answer: nucleotide excision repair, base excision repair, and mismatch repair all work together to maintain the integrity of DNA by identifying and correcting different types of errors and damage in the DNA. These repair mechanisms work at different stages of the cell cycle and use different enzymes and processes to repair different types of damage, but they all serve the same overall purpose of keeping the genetic information in the cell accurate and functional. Question: can you provide an example of a disease caused by a defect in one of the DNA repair mechanism? Answer: Xeroderma pigmentosum is a disease caused by a defect in nucleotide excision repair mechanism. Individuals with this disease have an inability to repair DNA pyrimidine dimers caused by UV exposure, which can lead to dry skin, photosensitivity, and an increased risk of skin cancer. Similarly, Lynch syndrome is a disease caused by a defect in the mismatch repair mechanism, which can lead to hereditary non-polyposis colorectal cancer.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the First Aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.